Hello, welcome to the Google Tech Talk. We are very pleased to have Professor Marc with us today. Let me give a brief introduction. He received his BSc degree in genetics from Autonomous University of Barcelona, Catalonia in 1994, and he continued there with a UAB scholarship and received his PhD in biophysics in 1999. His PhD was under the guidance of Professors Martin Karplus, Frances Evers, and Baldomero Oliva, and he focused on development of methods for folding studies using molecular dynamics. After that, he joined SALI Lab, which is a laboratory of molecular biophysics at Rockefeller University as Barav Wellcome Fund postdoctoral fellow, and he was a research associate at the same lab. There he concentrated on developing comparative protein structure modeling methods. Since 2003, Professor Marc has been an adjunct assistant professor at UCSF. Today, he's going to talk to us about tropical disease initiative, why it is interesting to Google, and in my opinion, is very important for the entire world. So please welcome, Professor.
Thank you, Akash, and thank you, guys, and also Avichal, who I know is in New York, and he's the guy who heard that very same talk at Stanford, where I gave it about a month ago or 2 months ago, and he invited me here. So I thank you all, both of you, guys. And as you heard, I'm a biologist, so don't expect me to talk too much about computational science or engineering. I'm a biologist. I did my degree in genetics. But then I moved into the theoretical biology, working on protein structure prediction. That's what we do, and that's what I call my day job. We do comparative modeling. What comparative modeling is we take all known protein sequences, and for you guys that are familiar with the genome, each gene produces one protein, and the protein is actually what makes the function, your cells function. So we take all those proteins and we try to predict the three-dimensional structure. And here's depicted. Each one of these little rows here represents one protein. And we try to do all the proteins that we get our hands on. This is in the framework of structural genomics. This is the next step after the genomics effort. And what we try to do here is to predict the structures so that you guys, we can actually work on developing new drugs. But this is my day job. This is what UCSF pays me for, and that's what pays my bills. But at night, I work on TDI. And unfortunately, UCSF is not as nice as Google, so I don't have my 20%. But this is my 20%. My 20% starts at 8 p.m. and finishes at 1 a.m. That's my 20%. So I work at TDI, and I'm going to be talking about TDI because I think TDI is more appropriate for you guys, because we are eager to welcome anybody, not just the Google people, but anybody to help us on reaching our goal, which is developing new tools that is going to help people, researchers out there, to develop new drugs for neglected diseases. There are tons of neglected diseases, mostly in Africa and India and China, and those neglected diseases are called neglected because the big pharma are not trying to do anything about them. We have also neglected diseases in the US, but they are neglected because they are affecting only a very small part of the population of the US. In Africa, what happens, they are neglected because people can actually not pay for the drugs that they would need. We're talking about malaria, Chagas disease, cholera, tuberculosis, etc. And it's not affecting a small number of people, it's affecting millions and millions and millions of people. So I think it's important to start developing some sort of open source biology so that we actually can do that cheaper, lower the cost of developing the drugs, and then therefore these people can access to those drugs. So I'm dedicating my 20% of the time to TDI. Some pointers for you guys. If you want to know a little bit more, you should go to tropicaldisease.org. That's the website for TDI. I also put here some flyers if you guys want to take them with you. We have two partnerships, one with the Nature publishers. They run our wiki site, and another one which is very good. It's with the people at the Synaptic Lab. This is a new organization. They are a nonprofit organization. It's led by Ginger Taylor. She used to work for PeopleSoft, then Oracle, and now she actually quit and decided to dedicate herself to developing whatever she's doing at the Synaptic Lab. Her goal is to generate tools for an open source biomedical research. So TDI is actually the first client that she's getting. She's going to be developing tools for us. And her goal is to accelerate the drug discovery process from basic research, that's what I do, to clinical research, that's what medical people do. So if you want to know a little bit more, just go into their website. So let me give you some, I'm probably gonna get this with me. Let me give you some pointers. Some time back ago, we started this about 2 years ago in 2004 by publishing a paper in a journal called the Public Library of Science Plus in Medicine. So we got that paper out and people were excited about it. We got tons of emails, and here I'm just picking some of them. Most of them will say, oh, this is great, but what the hell is happening here? What can I do for helping you? Another one would say, you guys look good, but you lack an action of plan. Then you have some of the people that say, if someone will tell me where I sign up, I'll give you some research topics to begin to, I'll be grateful. So the message was clear. We like your idea, but please help us to do something for you. This I like very much. Is a programmer, like you guys, most of you, I guess, are programmers. Not even a bioinformatician who got interested in what we're doing because probably saw the open source word somewhere and then he got interested. But he says, no, started with RMS. He gave us a programming administration tools to play with. Linux started with Linux, so we got a new operating system to play with. Please, you got to get somebody great in the field and release something for everybody to play with. And that's what we're actually trying to do. We're trying to get some people that are experts in the field of malaria research, schistomyosis research, tuberculosis research, etc., to start putting out there some things to do. And we try to actually provide the tools to those experts to play with the data. And this is one of the founders of the idea, Stephen Maurer. He's a lawyer at Berkeley, and he's the first author of that paper I mentioned before. He says that if we do science well, everything is going to come into place. And we hope that we actually start doing the science well. And I'm going to show you a few slides about the science that we do. So what the talk is going to be about is Tropical Disease Initiative. I'm going to show you what is TDI and how it can affect the drug discovery pipeline, what computational biology can do, what computational biology has done so far in the drug discovery framework. And finally, and probably the most interesting for you guys, I hope you do not abandon me here, is the projects that we are actually trying to develop and where we actually need help from engineers and programmers, projects within the TDI and TSL. So what is the pipeline for drug discovery? For an average drug out there in the market, it takes a very undefined time period. And it's undefined because these initial pre-steps are undefined. You can spend your whole life trying to improve that part here. But when you have some, what we call the leads for a drug, which is molecules that look like they're going to be good candidates for drug development, it takes about 12 years before it reaches the market. Okay? Now, the success rate, it goes from 100%, suppose that we've said the 100% here, it drops dramatically at the beginning and it gets very small. So you actually need a lot of these lead compounds to reach one drug in the market. And the cost skyrockets when you go into this step. So it's actually very important to two things. Lower the time that you spend in here and improve the accuracy of the leads because you want to actually have the success rate higher and the cost lower, because actually the cost is penalized because you try a lot of things with things that wouldn't work anyway. So how we do that, and that's the only part that TDI is interested on, the target lead identification. We combine computational tools and interactive biomedical chemistry, this is experiments, to predict what are the targets in your cells that are going to be interesting for a particular disease. Let's say cancer. We want to find all proteins in your cell that are affected by that particular cancer, and we want to tackle those. And we want to find inhibitors or leads or compounds that interact with that particular target. And that is done computationally speaking, and we have developed tons of tools for that. Then when we set an initial list of target inhibitor compounds, there is some chemistry that is going on to validate those hits, and so on and so forth. We go into circles, and that's why it takes so long, because you actually never reach a consensus until the point that, well, you have a list of probable leads, and then they are sent to lead optimization, preclinical, etc. So TDI, we hope is going to short the time here and increase the success rate, because TDI nowadays is possible for several reasons. It wasn't possible about 5-10 years ago. It's possible because we have completeness of genome projects. Only about half a year ago, they complete the malaria genome, which is now we have a whole list of parts of the malarian genome. So we can start working on the malaria at the genomic level, not just in particular genes. We have new and complete biological databases that we can mine, and we can actually subtract some information there. And if you guys would ever go into look in any of those biological databases, you probably would jump right away from the window, because we are biologists, we are not computational people, and we started those databases something like 30 years ago. If you look at those, they're still probably 20 years back in computational science. So it's pretty hard to actually work with databases of biological data. We have new software and computers which get cheaper and faster. We have internet we didn't have some time ago, at least as spread as it is today, which means more people looking at it, which means less cost of developing a new drug. The problems we're facing in TDI is that computational biology alone is not enough. We still need to do some experiments, and experiments are very expensive and very time cost. So there's still limitations, but we hope to overcome those limitations. This is the flow chart that I mentioned to you before, but with more details. We have databases of all type of information in biology: genome sequence, protein structure, ligand libraries, data mining from journals. It's very important. Not a lot of people is doing that, and hopefully the semantic web is going to help towards that. We have millions and millions and millions of texts, and the scholars, Google Scholar is doing a great job on that. But we want to associate each one of those particular entries in the library to genes and to proteins. And that's very difficult, because a given protein might have 30 different names. Okay, people have named it so differently. It's very difficult to find the proper links between the protein and the data in the literature. We have other databases, and then we have chemistry. What we hope is we're going to go around until we get some leads, and companies called virtual pharma are going to take those to the production and clinical, actually first preclinical, clinical research, and then production, and then they're going to distribute that into the third world. What are virtual pharma? It's a quite interesting approach, and it's really new. It's something about 5-10 years. It's people who have started a non-profit pharmaceutical company. So non-profit and pharmaceutical companies seem not to go along, but they actually do. And the first one is based here in the Bay Area. It's the One World Health Organization. And they actually are delivering, I think in a year or so, the first drug into an India village that they actually going to take the first drug, developed under the help of virtual pharma. They are non-profit, and what they do is they take money mostly from Bill Gates Foundation and NIH, and they do that so that they lower the cost of production of particular drugs. So we hope that we're going to be helpful to virtual pharma by getting them better leads so that they have higher success rate. So let's go into computational biology. And there are tons of tools that computational biologists have used to develop drugs. You can find a list of them here if you're interested. If any of you guys is really interested in bioinformatics, I encourage you to go through that particular list. So what are those tools available? and I'm going to stay here for lunch if you guys want to know a little bit more about bioinformatics itself, not just this open source biology that we're trying to do. I'm more than happy to talk to you guys later on about bioinformatics itself. But this is one thing that we're doing. We're trying to map all protein ligand universe, and ligands are small compounds. When you take a drug, you're actually taking this small compound into your body, and that small compound is going to interact with certain proteins in your body, and it's going to make you feel good or bad, depending of actually how is your genome. And for those of you that were here about in February, I think Russ Almond was here giving a talk, you probably heard about these things. So we're trying to map the universe of that. How we do that? By using, there are several groups involved in that, including myself. We take all known protein structures. We take all list of small ligands, and NIH at Washington has a very good public available list of drugs. We take all protein-protein interactions pipeline, and we apply our methods of prediction towards this pipeline, so that at the end, including a graphical user interface, at the end, a user, a biologist, could go there and have a whole map of the protein ligand universe. So he wants to know what a particular drug is interacting to a particular protein. He will go there and obtain all this information through that pipeline. This is the first project we're going to be implementing in TDI as the science part is. And we hope this is going to have a huge effect. We're going to probably start with a particular genome, let's say malaria or tuberculosis, which are of very much interest. And we're going to pipeline all those genomes through here and make all the data available. These are the databases for you guys who might be interested. There are tons of different databases. I distributed them in this way. We have sequence-based, functional-based, and structure-based. This is the world I work on, more or less, around here. But there are all these databases, and I don't know which one is scarier. But they are the databases, and this is what we have to deal with. It would be nice that actually Google or somebody indexes all those databases and creates a unique database with all the synonyms that connect between them and actually makes them available in an easy-to-search way. What we do is protein structure modeling, and we have a software that does it automatically. We take all the sequences at the beginning. We build what we call the profile of sequences so that we get all the evolutionary information for a particular sequence into our database. We scan for known structures, and then we build by comparative modeling the three-dimensional models, and we use that as an interface to our software. Every software we develop, we make an interface so that it's ready available for everybody to use. So what is comparative modeling is what goes into here, and what we do is we infer information. It is known in evolution that if two sequences are somehow similar, or what we call homologous, they're going to have probably a similar two-dimensional structure. So what we do is, if we know the structure of protein B, and we want to know the structure of protein A, and they are similar, we copy all the coordinates of structure B into the sequence of A, and that's the way we actually predict the three-dimensional structure. We're doing that in an automatic fashion. There is tons of algorithms behind that, which I'm happy to talk to you later on if you are interested on. We do that for all sequences in PDB, all sequences in the world. There are about 3 million sequences known for proteins. Everybody of us carries about 30,000 different sequences in our body. But there are tons of genomes out there, and microorganisms is probably the most extensive of them. There are about 3 million known sequences. We have models for about 1/3 of those. For you to contrast, experimentally speaking, what is known about the three-dimensional structure of sequences is only about 30,000. We know, experimentally speaking, only 30,000 structures, different structures for those sequences. So there is a huge gap between 3 million and 30,000. And that's what computational biology is trying to fulfill. And we actually doing a pretty good job. We are at 1/3 to completeness of the sequence space. When we have the three-dimensional structure, and here you can see one protein, particular protein, bound to a ligand. The ligand will be in yellow. And then in red are the residues of that particular protein that interact with the ligand. When we have that particular protein-ligand interaction, we can actually predict some things. And here is what I show you, that we have tools that are able to obtain information from the three-dimensional structure and predict which one of these ligands, and these are just the ID codes for different ligands. ATP, this is what is give you energy in the morning. So we take a protein and we ask ourselves, is it binding ATP or not? If it binds, where it binds. So if you do a random guess, you will get around 20% accuracy, which means that 80% of the time you run. But applying our algorithm, for example, for ATP, we reach at about 80% accuracy. So with our algorithm, we can take ATP and any given protein and try to figure out where in the protein that binds. And this is important because the next step would be to use a database of drugs and to find an inhibitor that binds into the same place that where ATP is binding, and therefore inhibiting the protein to do the function that it's trying to do. Imagine that that function is against your own will. You're having a cancer and you're trying to inhibit a protein that is giving you the cancer or the disease. So we want to inhibit it. So we go into databases like this one, a Zinc database developed by the group of Irwin and Schoikert at UCSF. That is a free database of commercially available drugs. What it means commercially available? It means that if you want to do an experiment, you can actually go into a company and buy the compound. They synthesize it for you, you get it in the lab, and then you try it. So it's important it is also available. Not all compounds are chemical, we can synthesize them chemically. So it's important it's available. So we take this database, and what we do is we take the structure. You remember that we have predicted 1/3 of all the structures in the PDB, in the universe. We take and now we put it in the computer together with the database of ligands, and we say, I tell the computer that this is my binding site where the M is, okay? My compounds are going to be somewhere binding here, and I want to inhibit that binding site. I take all compounds, run it through the computer, and that usually takes a lot of time, and then get some hits that I'm going to put into experimental work and determine. So this is done computationally speaking. Experimentally speaking, it's also done, and it's called high throughput screening, and pharmaceutical companies do that, but it's extremely expensive. Okay, so computational time speaking, we save time and money. For example, there was this protein in tuberculosis that we wanted to inhibit, a reductase. This is just a name of a protein. It does a particular function. We took a particular library, and we find hits which are significantly inhibiting the protein. If you do high throughput screening, experimental, you get only about 2% of the hits hitting that particular protein and inhibiting it. In docking, we find about 6%. So the docking is not only faster and cheaper, but it's also sometimes, this is a very good case, sometimes it finds hits that high throughput screening couldn't find. And I'll just skip that slide. This is telling you the same story. It's not a single case that we have been able to do, but we have lots of different proteins that are affecting different diseases in which we have used different methods of docking, and they were good enough to find a particular ligand. And then those ligands have to be optimized, and then they have to go to preclinical, clinical, etc. Those are steps that I'm not going to talk about because we do only basic research, not clinical research. Also, one thing that is important, a lot of these drugs, they are very good in vitro. But when you take them orally, your stomach dissolves them, and therefore it's not doing any good to you. Or sometimes you take them, and basically is affecting, the secondary effects are even worse than the disease itself. So not everything looks as good as it is, but we are advancing in that. This is what we have done so far using computer. Yes?
What determines the complexity of finding a docking and making a match, and how does it affect the error rate?
So your question was...
What is the inherent difficulties?
What are the most inherent difficulties about doing that algorithmic docking, this in silico docking? Well, most of the problem is that we are talking about a very, very detailed representation of the system. We're talking that if you move an atom an atomic position for 0.1 angstroms away, you might lose some certain interaction and your energy might go skyrocketing. And therefore, it's very difficult to pick up these little subtle changes in your energy function. Because what you do is you try a lot of different positions for the atoms, pair each one of those ligands, you try a lot of different positions, and your energy function, it has to say, this one here is better than this one here, but just a slight movement might change your energy function. So the limitation that we're facing is two. Energy function, meaning that the scoring functions are not perfect, and the sampling, because you have to sample so much. Computationally speaking, it's expensive. And each one of these experiments might take months of CPUs. So we're facing both sampling and energy scoring.
And a related question. So I give you two proteins and two ligands, and you want to figure out which one is easier to match than the other. Can you compare them? Can you predict that which one will be?
So what we do is we actually take a library of ligands and dock again a particular protein and tell you which ligand is the most probable to be inhibiting that particular protein, but we can also compare two other proteins. We can take now two proteins and dock the same ligand and see which one of those are the most probable to be affecting, to be docking the particular drug. So for you to get an idea of what is happening, the first hit that used what we call structure-based drug design, meaning that they took the structures, they look at the structures, they use computational tools, and they predict certain inhibitors, was at the end of the 80s with the HIV proteinase inhibitor. At the end of the 80s, HIV was hot. Everybody was actually researching on HIV. Everybody was actually looking to find some inhibitors for that particular proteinase, and the group of Blundell in England were one of the first to use knowledge-based protein modeling and design, and they actually came out with good candidates, and they are, some of those candidates, they are now in the market. So the things are happening. More things that recent, 2003, we had the SARS problem. It was hot 2003. Nowadays, we're not talking too much about it because there are not more new cases, but who knows what is going to happen. These people, Lindaski and colleagues, took the server. They have a server which is quite interesting. It takes predictions from all other servers around the world that they do predictions about protein structure. And then they collect that prediction. It's what we call meta servers. They collect that predictions and they make a consensus prediction. We know that methods A, B, and C might be wrong, but not everybody is wrong at the same time and the same problem. So the consensus actually gave them access to a particular prediction that nobody else have done, which was interesting enough to be published in one of the top journals papers, the Cell journal, as a possible target for drug development against the SARS. So it's an interesting approach. It's done only computationally speaking, and these people have used methods that they didn't develop to combine the final information. These are actually the methods they used. Experimental approach. This is also very nice work done by Joe DeRisi group at UCSF. What they do here, they take malaria genome and they actually go into a cycle. The malaria cycle, when it's inside your blood cell, it goes to a particular cycle which is very consistent. All the time it's doing exactly the same things. It doesn't matter what happens to the surroundings. Malaria is strong enough to actually do all those steps in a given 24 hours. Sorry, 48 hours. So what they did, they studied all the genes. Each line of here represents one gene of the genome. And they studied the expression of the gene. In that way, they could know at hour 6 of the life of malaria in this blood cell, genome A was overexpressed and genome B was underexpressed. What it means, I know what genome A is doing, so I can now guess what the cell might be doing, okay? They found something which is extremely surprising and never seen before, is that there is a regular pattern. You see, green, it means overexpressed, red means underexpressed. It says that the genome is regulated, completely regulated. The expression of the genes follows a cascade. When A is on, B is off, etc., etc., etc. So there is a cascade, which means that that particular organism is extremely strong. It doesn't matter what the environment is, he's going to do what he has to do. So it's going to be difficult to find a drug to actually disturb that particular cascade of expression. So it's a very nice piece of work. They have a database with all this information available, which now somebody has to go and data mine. So there are drugs out there for companies that are based on drug development, on structure-based drug development. Informatica, they have drugs that are almost going to optimization of leads. Vertex, they have even drugs out there. Let me see if, yeah, they have 2 drugs out of the market. Predix, etc., you name it, SGX. Lots of these companies around here in the Bay Area, biotech, they actually started using this type of approach. So we reached the final part, which is the TDI again. I've talked to you about the science that we're doing in TDI, but now I'm going to talk to you the particular projects that we have envisioned for TDI, and I hope some of you guys might be interested on giving some of this 20% of your time into helping TDI to move forward. So these are the projects. There are 5 projects. In green means that we are in advanced stage of the project. Actually, the structure prediction is already done. And in orange means that we're thinking about how we actually know how to do them, but we still don't have anything to show. And then the gene basket is something that I hope you guys are going to like. It's something that we're still thinking about how to do. And this is all in collaboration with the Synaptic League people. So gene cards, what we do here is, I told you, there are tons of databases about particular information that people know about proteins, ligands, and sequences, and function, etc. So what we have done is we have collected all this information from these 3 different databases. Biomart is already a compendium of several databases. So it's not only 3, but it's several of them. NCBI, this is NIH, the National Institute of Health, run by the government of the US, has also a lot of different information from different databases they have compiled and put it on one site. So what we try to do is we take all the genes by malaria, let's say, in that particular time, malaria has been defined that it has about 5,200 different genes. And these numbers always change. That's the way it is, because we are not good enough to predict the exact number of genes in a particular genome. So we take that and we collect all the information about literature. We try to make this matching between literature and genes, annotation, functional annotation, structural information, and function. And we give, this is a collection of links, so when somebody goes into our website, it's going to be able to find a gene card of all the information. The next step we want to do is we want to move scientists, which is very difficult, into the 21st century tools. It sounds strange, but scientists do not use wiki and blog, okay? We do not, because that's not the way we were showed that we could share our information. Our information is shared when we publish something in a paper. So we're trying to actually encourage people to start using wiki and blogs. And we use Drupal, so we have the blogging module, but we also want to develop a wiki that takes that static information that we have now and lets people to start not modifying, because this is actually experimentally verified, but adding more information per gene. And that is something that we want to develop. And about 2 or 3 months ago in Nature, there was a huge discussion about, there were 3 or 4 articles about the new technologies that scientists are not using. And there was an open public call for a wiki based on gene information. And we hope somebody's going to take that call and actually develop a wiki-based gene information. I told you that we have done this protein structure modeling. This is the second project, and it's completed because we actually have this pipeline software that we just press a button and predict all the structures of the genome. We took the 5,270 genes of malaria, And we have been able to build three-dimensional structures for about 2/3 of the database of malaria genes. This is the total number of models, which is always higher than the sequences because sequences have several domains and so on, but if you are interested in these particulars, I can talk to you. The next one is also interesting. It's what I call a voting system. There is plenty of people who knows a lot about malaria, okay? But we cannot reach them one-to-one. What we want to do is to put out there a voting system, what we call target selection. It's a fancy word to say voting system. What we're going to put out there is all the genes of malaria with the gene cards. People can look at that, and then we're going to allow users to post questions such as, which is the most probable sequence or protein that is going to bind to that particular drug? Please vote. And experts out there are going to go and vote for a particular gene and rank those genes given the voting, okay? So that's what we're trying to do for structural genomics. There is this professor, Raymond Hu, at the University of Toronto, who works on malaria, and he tries to predict all the structures of malaria. His group is dedicated to malaria research. But he wants to know which ones of all the 5,270 genes is the most interesting one to predict the structure. So we're going to allow people to vote and tell Raymond, even if they never actually met, tell Raymond, this is a very important protein. Please determine the structure, because if you actually determine the structure, I'm going to do X and Y experiments on it, and we hope to find a drug for that particular structure. So this is our first pilot project for target selection. And we are still lacking of the software, the interface software for people to actually vote, because it's not just a voting system. You have to be sure that the people actually are putting the right information in there. So we have to put some filterings. This is the wiki gene. I already told you. We want to modify that particular gene cards by experts in the field, and these are actual experts in the field to put their input. And this is the gene basket. What we're trying to do here is similar to Yahoo Flickrs or Delicious are doing. And we want to do it for genes. Let's imagine that this is my set of genes, okay? And the information that we have nowadays is like this. You have a set of genes that they do have some names on it. Some of them have a name. Some others do not have a name. But what you can do actually is you can start comparing them. If you compare them, there are tools out there to compare genes. You're going to know that oranges and oranges are similar, and they have the same shape as apples, but they are not similar at all than bananas, but they actually are fruits, all of them. Some of them, you're going to find that they are identical, so you just put them together. And some of them, you're going to find that this particular brand of chips goes with this particular brand of chips. For some others, you cannot say anything. It's just a gene. You don't know anything about it. So the next thing we're going to do, we're going to allow people to actually put genes into their baskets and tag them and give some information about them. They can go into internet, browse the internet, get into NCBI website for literature, and click into a bookmarklet that they will bring their basket up. And it's going to ask them, okay, you just want to put that particular link or information into this particular basket. Which gene do you want to associate it to? And then you click gene A or B to associate that information. This is very powerful, I think, because what we have done, biologists have done now so far, is automatically we have generated algorithms that go in there, search for literature, and try to associate automatically the literature or the pieces of information to genes. This is powerful, but it has a lot of false positives because the problem of the synonyms. Not all proteins are named the same way, and several proteins have several names. So it's a lot of false positive. In here, we're trying to take the workforce of people and ask them physically to voluntarily click and associate things. So now what we're going to have is that we're going to have things associated, apples with apples, oranges with oranges. But not only that, if they start putting tags, we know that these are fruits. There is automatic connection. Before we knew that these two baskets had the same shape, but now we also know that bananas actually are fruits as well as apples, and we can associate them this way. And not only that, they might have the same pieces of information that the people have found out there in the internet associated to different genes. And that's a link that didn't exist before anywhere. Not only that, now we go and we ask ourselves, who is actually working on that particular basket or the genes in that particular basket? We have two experts. They know each other. They actually work in the same problem. They probably know each other. But the third one, they didn't know that he or she was working in that particular gene. So they can now use the tools that we have in Drupal to communicate to each other and start talking about science, which is important. We have to make the life of scientists easier to work with this huge amount of data that is out there in the databases of biology. So this is our gene basket. We're still thinking about how to do that, what best to do in that. And if any of you guys is interested in developing the tools behind that, we will be more than happy to welcome you. This is something that just came out this week. This is this week's volume of Nature. In Nature, you can actually write a letter to the editor and they publish it. And you ask for something, and Marc is at Yale. He's one of the leaders in this country for bioinformatics. And I didn't post the initial part, but the initial part started saying Google Earth has been very good. We have now a tool to go around the world from point A to point B, and we know what we have to do to go from point A to point B. And not only that, we know what we're crossing by, and we know that in this corner there is a pharmacy store, and the other corner there is X and Y. Why don't we have that for biological science maps? So it would be extremely nice to have a sort of flexible Google X map that allows scientists to actually produce coordinates for you guys, and then we can actually go and browse it. And let's say that I'm talking about a particular protein, three-dimensional structure of a protein, and I want to know how to go from residue A to residue B towards a channel that has specific properties. And it would be nice to just click once and find this channel, because this is something that biologists do not have, and it would be extremely, extremely, extremely useful. And I'm just giving you an example of a protein, but let's say that you want to go from point A in the cell to point B in the cell, going towards passing through the ribosome and going to a particular compartment in the cell. That would be beautiful to have. And this is something that Marc has asked in the latest Nature. So any takers here? Finally, what you can do for TDI is that we have to put this concept of the power of us, which means the collaboration and freely giving away all your information so that the rest of the people can actually use it. So that sometimes it's very individualist, biomed research community gets not that individualist, and so we need collaborative tools, and that's what the TSL people, the synaptic leap people are trying to do. We want to reach worldwide. We need exposure, and I don't know, you work on the search engine. It would be nice if somebody types malaria, TDI goes into the top. Enhance research. We need to port existing biocomputing tools into the internet, and this is something that biologists have always struggled, because we are biologists at the end of the day, so it's difficult for us to actually get it right when we put things out there on the internet for everybody to use. If you are a researcher or somebody that is just interested in it, if you join, you will be part of an innovative new approach of research. Nobody has done open source research so far. This is something that people start talking about. You're going to be able to connect to peers that are interested in the same problems, and you're going to be able to develop new research tools that didn't exist before. And by research tool, I mean biological research tools. Somebody's going to do biology, real biology, using the tools that you developed. And of course, it gives good karma to everybody. So I always like to finish my talk with that slide, and it's what I call the 595%. 95% are the clients of TDI. It's 95% of biologists out there who has no idea, they don't have any idea of doing bioinformatics, and they would love to have an easy-to-use interface that gives them access to all the information and collaboration tools. And these are the ones that we're lacking so far, because the 5%, which is me including, and if you guys volunteer, probably you're going to end up in this 5%, are the developers who have to do the work now to develop those tools so that these 95% of people actually can do science using the TDI tools. So I think we're doing a good progress here, and as anything in open source and collaborative communities, nowadays we have a plateau that we hope it's going to explode at some point, and we're going to get all those volunteers to do the science at some point. And we're feeling that we are pretty close to that. We have about 50 people actively join the TDI TSL site, but at any point of time we have in between 30 and 40 people visiting the website, which is not bad for something that has been out there for only 4 months now. So let me finish by acknowledging the people in our group who has actually done some of the research I've shown you today. The people involved in the Tropical Disease Initiative, which is myself, Stephen Maurer, Arthur Rai, Andrei Shally, Thomas Kleber, and Matt Todd. The guys at the Synaptic led by Ginger Taylor and Randy, Chris, Rick, and Ken. And the people at the Nature Publisher Group, Timo Hane and Declan Butler, who are running our Wiki site. And I encourage you to read more in these 2 articles. And I have a copy of this one right here if you want to take one, as well as 2 flyers for the TDI and TSL websites. And you can go to this Nature volume 438, in which there are those articles talking about scientists using blogs and wikis and moving towards the, more collaborative efforts. So it's an interesting to read, and I think you guys will be interested to read that particular entry in Nature. If you want more information about the particular research I do, you can go here, or for the tropical disease or the synaptic lip, you can go here, and now I'll take any questions you might have. Thank you. So we don't have anything of the human genome in any of those. Oh yeah, I have to repeat the question. So the question was, how much of the human genome is covered by the gene cards or the gene basket? We only have the malaria genome in the gene cards so far. But you can go to NCBI website of the National Institute of Health, and there you have all the human genome parsed and compiled. We are focusing only on neglected diseases, and for now we're doing all the genomes for for parasites or organisms that produce that neglected disease in particular. Gene basket doesn't exist, it's only an idea so far. Okay.
Can you talk a little more about like how gene cards differ from like NCBI's website, for example, or like Contract, or just like these other websites that bring together the papers and the annotations and the sequences?
Yeah, the question was how gene cards differentiate from other existing databases that collect this information also in a concise manner, I believe. So the difference is that we are focusing on a particular genomes, one of the difference, but that wouldn't be enough. I think it's just a matter of putting a card. A card doesn't contain all the information, just the links. And I could go back, but it doesn't make a difference. We collect links. We are not duplicating the data, we're just collecting links, and we're trying to find the synonyms between the databases. So a protein A might be named 8' in another database, and we have to make a link. That doesn't exist for all the databases that we collect the links, because we collect links from different databases. The point here is that the user of TDI TSL will be able to see at a glance all the information available for a particular gene before it makes a comment or a vote. So the additional value of TDI will be those tools that do not exist nowadays yet. Okay, thank you very much.